Hello students, today I bring for you a new chapter and that is microbes in human welfare. It is a easy chapter and important one. In this chapter, we are going to cover topics like microbes in household products, microbes in industrial products, microbes in sewage treatment, microbes in biogas production, microbes as biocontrol agents and microbes as biofertilizer. We will cover this PDF as well as the NCERT. So let's start. First of all, various type of microbes. Here you can see bacteria. Rod-shaped bacteria magnified 1500x. Then bacteria spherical shaped magnified 1500x and a rod-shaped bacterium showing flagella. You can see. Then a bacteriophage and adenovirus which causes respiratory infections. Questions are asked about this. And rod-shaped tobacco mosaic virus. You can see the structure. Here again some more diagrams. Colonies of bacteria growing in a petri dish. Fungal colony growing in a petri dish. First of all, microbes in household products. Lactobacillus or lactic acid bacteria, LAB. It converts milk to curd by producing acids that coagulate and partially digest the milk proteins. Fresh milk can be converted to curd by adding some curd containing LAB. It also increases vitamin B12. In stomach, LAB helps to check pathogens. Then bacterial fermentation, anaerobic respiration in dough is used to make food such as dosa and idli etc. Puffed up appearance of dough is due to the production of CO2 gas. Then baker's yeast, Saccharomyces cerevisiae. It is used to make bread by fermenting dough. Toddy is made by fermenting sap from palms. Microbes are used to ferment fish, soybean and bamboo shoots and to produce cheese. Here, Swiss cheese has large holes due to production of CO2 by Propionibacterium sermani, a bacterium. And Roquefort cheese is ripened by growing a fungus, Penicillium roqueforti, on them. You can see in the diagrams. Then microbe in industrial products. Let's see your NCERT what it says about this chapter. Here you can see. First question is asked, which kingdom among the living organisms contain microorganisms which are ones that are only microscopic? And here the answer, microorganisms are found in the following kingdom. Kingdom Monera contains microscopic prokaryotes, that is bacteria. Kingdom Protista includes unicellular eukaryotes like amoeba, fungi, bacteria and other microorganisms. And Kingdom Fungi, only yeast is microscopic. Here, the diagrams we already seen in that PDF. As I told you, a question is asked, adenovirus which causes respiratory infections. Remember. Then, microbes in household products. Common example, curd from milk, microorganisms such as lactobacillus and other commonly called lactic acid bacteria, LAB, growing in milk and converted into curd. During growth, LAB produce acids that coagulate and partially digest milk proteins. A small amount of curd is added to the fresh milk as inoculum or starter containing millions of LAB which at suitable temperature multiply, thus converting milk to curd, which also improves its nutritional quality by increasing vitamin B12. In our stomach too, LAB play a very beneficial role in checking disease-causing microbes, that is pathogens. Then the dough which is used for making food such as dosa and idli is also fermented by bacteria. The puffed up appearance of dough is due to the production of CO2 gas. And here, question is asked, can you tell which metabolic pathway is taking place resulting in the formation of CO2? Answer will be aerobic respiration. Then, similarly, the dough which is used for making bread is fermented using baker's yeast, that is Saccharomyces cerevisiae. and about toady also mentioned. Then Swiss cheese are due to production of large amount of CO2 by bacterium named Propionibacterium sermani, and the Roquefort cheese are ripened by growing a specific fungi on them. Then microbes in industrial products. Let's see the PDF here. Production of beverages, antibiotics, etc. on an industrial scale requires growing microbes in very large vessels that are called fermenter. You can see how large are they comparing to a human being. These are called fermenters. Saccharomyces cerevisiae, brewer's yeast is used in the production of beverages by fermenting malted cereals and fruit juice to produce ethanol. Wine and beer are produced without distillation. Whiskey, brandy, rum, gin and arak etc. are produced by distillation of fermented broth. Then antibiotics. Antibiotics are chemical substances produced by some microbes and can kill or retard the growth of the pathogens. Used to treat plague, whooping cough, diphtheria, leprosy, etc. Penicillin. It is the first antibiotic discovered by Alexander Fleming. He observed that Staphylococcus could not grow around a mold, that is Penicillium notatum, growing in unwashed culture plates. He extracted penicillin from it. Then Ernest Chain and Howard Florey established its full potential as an effective antibiotic. Fleming, Chain and Florey were awarded Nobel Prize in 1945. Then chemicals, enzymes and other bioactive molecules. Organic acids. Acid producer microbe includes microbes are Aspergillus niger, a fungus, and it produces acid, citric acid. Acidobacter aceti, it produces acetic acid. Clostridium butylicum, it produces butyric acid. Lactobacillus, it produces lactic acid. Then alcohol. Yeast, that is Saccharomyces cerevisiae, is used to produce ethanol. Enzymes. Lipase, used in detergent formulation, helps to remove oil stain from laundry. Pectinase and protease, to clarify bottled juices. Then streptokinase, produced by Streptococcus, used as a clot buster to remove clots from the blood vessels of patients who have myocardial infarction. Then some more chemicals, enzymes or bioactive molecules are cyclosporin A. It is produced by Trichoderma falciparum, a fungus, used as an immunosuppressive agent in organ transplant patients. Statin, produced by Monascus purpureus, a yeast, used as blood cholesterol lowering agent. It inhibits the enzymes responsible for synthesis of cholesterol. Then next one, microbes in sewage treatment. Let's see NCERT what it says here. Beverages and antibiotics are some examples in industrial products, and production of an industrial scale requires growing microbes in a very large vessel that are called fermenters. See, diagrams are also here. Yeast have been used time immemorial for the production of beverages like wine, beer, whiskey, brandy or rum. For this purpose, same yeast, Saccharomyces cerevisiae, used for bread making and commonly called brewer's yeast, also used for fermenting malted cereal and fruit juices to produce ethanol. Then wine and beer are produced without distillation, whereas whiskey, brandy and rum are produced by distillation of the fermented broth. Coming to antibiotics, anti is a Greek word that means against and bio means life. Together they mean against life. Here Alexander Fleming, while working on Staphylococcus bacteria, once observed a mold growing in one of his unwashed culture plate around which Staphylococcus could not grow. He found out that it was due to a chemical produced by the mold and he named it penicillin after the mold Penicillium notatum. Then some more scientists came, Ernest Chain and Howard Florey. This antibiotic was extensively used to treat American soldiers wounded in World War II. Fleming, Chain and Fleury were awarded the Nobel Prize in 1945 for this discovery. Then it also treats deadly diseases such as plague, whooping cough, kali khasi, diphtheria, galgotu and leprosy, kushtrog, which is to kill millions all over the globe. Now coming to chemicals, enzymes and other bioactive molecules. Aspergillus niger, a fungus of citric acid, Acidobacter aceti, a bacterium of acetic acid, Clostridium butylicum, a bacterium of butyric acid, and Lactobacillus, also a bacterium of lactic acid. Yeast Saccharomyces cerevisiae is used for commercial production of ethanol. Lipases are used in detergents formulation and are helpful in removing oil stains from the laundry. And also here we all notice that the bottled fruit juices bought from the market are clearer as compared to those made at home. This is because the bottled juice are clarified by the use of pectinase and protease. Streptokinase produced by the bacterium Streptococcus and modified by genetic engineering is used as a clot buster for removing clots from the blood vessels of patients who have undergone myocardial infarction leading to heart attack. And another bioactive molecule, cyclosporin A, that is used as an immunosuppressive agent in organ transplant patients, is produced by the fungus Trichoderma falciparum. Statins produced by the yeast Monascus purpureus have been commercialized as blood cholesterol lowering agents. Then next topic, microbes in sewage treatment. Let's see the PDF here. Sewage, that is municipal wastewater, contains a large amount of organic matters and microbes. Sewage is treated in sewage treatment plant STPs to make it less polluting. It includes two stages: primary treatment, secondary treatment or biological treatment. First of all, primary treatment. Here you can see the diagram and the lines written in the PDF. It is the physical removal of particles. First of all, it includes removal of floating debris by sequential filtration. Sequential filtration. This is called sequential filtration. Removal of the grit, soil and small pebbles by sedimentation. Here you can see sediments are present here. That is removal of grit, soil and small pebbles by sedimentation. The settled solid form primary sludge and the supernatant form the primary effluent. Then secondary or biological treatment. Primary effluent is passed into large aeration tank and constantly agitated. You can see aeration tank and agitation occur. Okay. This allows vigorous growth of the useful aerobic microbes into floc, bacteria associated with fungal filaments to form mesh-like structure, that is called floc. Here it is mentioned mesh-like masses of bacteria and fungal filament, that is called flux. These microbes consume the organic matter in the effluent. This reduces the BOD, that is biochemical oxygen demand of the effluent. Then BOD means amount of oxygen consumed by bacteria to oxidize all the organic matter in 1 liter of water. It is measured of organic matter present in the water. The greater the BOD, more is its polluting potential. Then the effluent is then passed into a settling tank where the bacterial flocks are sediment. This sediment is called activated sludge. See the diagram. A small part of activated sludge is pumped back into the aeration tank to serve as the inoculum. Small part comes and serves as inoculum or starter. Then the remaining sludge is pumped into large tank called anaerobic sludge digester. Here some anaerobic bacteria digest the bacteria and fungi in the sludge by producing gases like CH4, H2S and CO2. These gases forms the biogas: methane, hydrogen sulfide, carbon dioxide. The effluent is released into natural water bodies like rivers and streams. Then here is the overall all step. Primary effluent goes to aeration tank, then constant agitation, then aerobic bacteria in flocks consume organic matter, then BOD reduces. Here you can see then it comes to effluent to settling tank, then flock sedimented to activity sludge, anaerobic sludge digester, then anaerobic bacteria digester sludge, biogas formed, and the effluent goes to the natural water bodies. Here again more diagrams. Next, the Ministry of Environment and Forest has initiated Ganga Action Plan and Human Action Plan to save from water pollution. Then microbes in the production of biogas. Let's see NCERT what it says. Here municipal wastewater is called sewage. Two stages: primary treatment, that is physical removal of particles large and small through filtration and sedimentation. Then primary sludge is also mentioned, you can see. Then secondary treatment or biological treatment. Here the flock, masses of bacteria associated with fungal filaments to form mesh-like structure is called flock. Then biological oxygen demand. BOD refers to the amount of the oxygen that would be consumed if all the organic matters in 1 liter of water were oxidized by bacteria. Then activated sludge. A small part of the activated sludge is pumped back into the aeration tank to serve as an inoculum or starter. Then remaining major part goes to anaerobic sludge digester, and here some more bacteria grows anaerobically. They digest the bacteria and fungi in the sludge, and it produces mixture of gases such as methane, hydrogen sulfide and carbon dioxide, commonly called biogas. Then you can see the Ministry of Environment and Forest has initiated Ganga Action Plan and Human Action Plan to save the major rivers of our country from pollution. Then production of biogas. Let's see the PDF. Microbes in the production of biogas. Here, biogas is a mixture of gases, mainly methane, produced by the microbes such as methanogens. Methanogens grow anaerobically on cellulosic material and produce methane along with CO2 and H2. Example, Methanobacterium. Methanobacterium is found in the anaerobic sludge and the rumen of the cattle for cellulose digestion. Biogas is used for cooking and lighting. Cattle dung, gobar contains these bacteria, so dung is used to produce biogas, that is gobar gas. Here the diagram or parts of a biogas plant. First of all, a concrete tank 10 to 15 feet deep to collect bio waste and slurry of the dung. A floating cover is placed over the slurry which keeps on rising as the biogas is produced. Second is outlet to supply biogas, and third is outlet to remove spent slurry, that is used as fertilizer. Then Indian Agricultural Research Institute IARI and Khadi and Village Industries Commission KVIC developed a technique of biogas production in India. Then microbes as biocontrol agents. Let's see the NCERT. Here again same points. Biogas is a mixture of gases containing predominantly methane produced by the microbial activity and which may be used as fuel. And here methanogens are mentioned, common bacterium, Methanobacterium. This bacteria also present in the rumen or a part of stomach of the cattle. They break down cellulose, and this is the diagram you can see. Dung and water goes to this, then gases are produced to gas holder, gas comes out and the sludge comes out. The technology of biogas production was developed by IARI, that is Indian Agricultural Research Institute, and KVIC, Khadi and Village Industry Commission. Then biocontrol agents. Here Biocontrol is the use of biological methods for controlling plant diseases and pests. Example, ladybird. Beetle that controls aphids. Dragonflies controls mosquitoes. Chemical pesticides and insecticides kill both useful and harmful organisms and causes pollution. Biocontrol methods has no such problems. First of all, agents are first Bacillus thuringiensis, shortcutly called BT, to control butterfly caterpillar. The dried spore of the BT available in the sachet in the market are mixed with water and sprayed on the vulnerable plants such as brassicas and fruit trees. These are eaten by the caterpillar. In their gut, the toxin is released and the larva gets killed. Scientists have introduced Bacillus thuringiensis toxin genes into plant, example BT cotton. Then second, Trichoderma is a fungus. They are seen in the roots ecosystem. They control several plant pathogens. Third, baculoviruses, especially genus Nucleopolyhedrovirus, attack insect and other arthropods. It is suitable for species specific, narrow spectrum insecticidal applications and desirable in IPM, that is integrated pest management program to conserve beneficial insects. See the diagram. Then microbes in biofertilizer. Let's see the NCERT. Here, same important points are mentioned. Biological control of the pest and disease, and beetles with red and black marking, the ladybird and dragonflies are used to get rid of the aphids and mosquitoes respectively. And another microbial biocontrol agent that is control butterfly caterpillars is the bacteria Bacillus thuringiensis. And toxin genes into plants such as BT cotton is an example. We will know about this type of BT cotton plants in chapter 10. Biological control being developed for use in the treatment of the plant disease in the fungus Trichoderma. Trichoderma species are free-living fungi that are very common in the root ecosystems. They are effective biocontrol agents of several plant pathogens. Baculoviruses are pathogens that attack insects and the other arthropods, and they come from the genus Nucleopolyhedrovirus. These viruses are species specific, means they only attack that particular insect, only the particular insect, and narrow spectrum, not a wider spectrum. And full form will ask the IPM, integrated pest management. Then microbes as biofertilizer. Let's see the PDF here. Biofertilizers are organisms that enrich the nutrients quality of the soil. Example, bacteria, fungus, cyanobacteria, etc. Rhizobium is a symbiotic bacteria in root nodules of leguminous plant which fix atmospheric nitrogen. Free-living bacteria in the soil, example, Azospirillum and Azotobacter, enrich the nitrogen content of the soil. Then mycorrhiza, symbiotic association of the fungus, example glomus, with plants. The fungus gets food from the plant. The fungal symbiont perform the following, that is, absorb phosphorus from the soil and passes it to the plant. Second, give resistance to root-borne pathogens and tolerance to salinity and drought. Third, give overall increase in plant growth and development. Cyanobacteria, blue-green algae, autotrophic microbes, they fix atmospheric nitrogen, example Anabaena, nostoc, oscillatoria, etc. In paddy fields, cyanobacteria serve as biofertilizers. It also adds organic matter to the soil and increase its fertility. Let's see the NCERT. Here, organic farming that uses biofertilizer. Biofertilizer are organisms that enrich the nutrient quality of the soil. Bacteria, fungus, cyanobacteria. And here we all know that the nodules of the root of the leguminous plant formed by the symbiotic association of the rhizobium. This bacteria fix atmospheric nitrogen into the organic form, which is used by the plant as nutrients. Other bacteria can fix atmospheric nitrogen while free living in the soil. Example, Azospirillum and Azotobacter. Then fungi are also known to form symbiotic association with plants, that is mycorrhiza. Many members of the genus Glomus form mycorrhiza. Remember, the fungal symbiont in these associations absorb phosphorus from the soil and pass it to the plant. Plant having such association shows other benefits also, such as resistance to root-borne pathogens, tolerance to salinity and drought, and an overall increase in plant growth and development. Here, cyanobacteria are autotrophic microbes widely distributed in aquatic and terrestrial environments, many of which can fix atmospheric nitrogen. Example, Anabena, Nostoc, Oscillatoria. In paddy fields, cyanobacteria serve as important biofertilizer. Blue-green algae also add organic matters to the soil and increase its fertility. Currently in our country, a number of biofertilizers are available commercially in the market, and farmers use these regularly in their field to replenish soil nutrients and to reduce dependence on chemical fertilizer. And you can see with this we have completed the whole chapter. And go through line by line of the NCERT and see this video. If you find it is helpful, the diagrams, then give a like. Do not forget to comment, and new viewers, please subscribe my channel. Thank you. Jai Jagannath.